So we're delighted to have Professor Rafi Malach with us today. Professor Malach, or Rafi, may I call you that, has done his postdoc at Anna Gradel's lab at MIT. She's an anatomist. He did his PhD before that in Berkeley, UC Berkeley, about visual plasticity. Since then, he's been at the Weizmann Institute of Science. He's been investigating vision and also awareness. He published groundbreaking papers, starting with revealing the human homologue of TE and IT cortex, which is the lateral occipital complex, which then his students in his lab proceeded to find exciting findings about these high order visual cortex regions. He's done groundbreaking research in moving into uncontrolled visual experiments with cinematic investigations, which was published in Science. He also published and found exciting findings about the tight coupling between neural activity and MRI signals. He also found revealing and exciting findings about memory-related activity in single unit recordings. He published in numerous top journals such as Science, Nature journals, and others. His students now hold positions in top universities worldwide. And we are delighted to have you, Rafi, with us to discuss vision again, going back to vision, and welcome to our seminar.
Great. Thanks, Sharon, for the very kind introduction. I will, before I start, I want some logistics points. I would like to present my presentation in full with no interruption for discussion, but I encourage you to interrupt me if something that I say is not clear. There's no point in me rushing ahead without my points being really understood. Another thing is that I will, in the interest of time, I'll only talk about minimal technical details. So if you have questions about technical details, please ask me in the question period. So, so let's start. The general topic I would like to discuss today is the neuronal mechanism that underlies the appearance of a visual image in our mind. What are the neuronal principles that cause us to see something? Very basic and very fundamental question. Now, if we are interested in... I suggest to mute. Okay. Please mute yourself because there are all kind of... Sharon, I think you can mute everybody if you want.
Yes, I can and I will.
Okay, so since I'm talking about looking for neuronal mechanisms underlying perception, I want to describe in a very quick overview, just a reminder of the techniques that we use in our group.
I have not at least.
The techniques that we use in our group to measure neuronal activity from the human brain. We are using non-invasive fMRI. I hope there's no need to elaborate on it. It's a non-invasive method that gives you activity signals from the entire brain. But the problem with this technique is that the time resolution is very poor. The signals are very sluggish. We use two other techniques that are very powerful, but very rare and very unique. These are invasive methods that are used in patients that undergo clinical diagnosis for epilepsy. We, of course, do not conduct the experiment. We are just taking the opportunity of the situation where a neurosurgeon is implanting electrode inside the cerebral cortex and the hippocampus of these patients. So this gives opportunity when they are off the clinical diagnosis to conduct studies of activity directly recorded from the human brain of either groups of neurons using contacts, which I'll call IEEG. These are groups of about 100,000 up to even million neurons in one contact. And also in very rare cases, we might be able to pick up individual neuron activity from the the cerebral cortex and the hippocampus of these patients. So again, before I dive into the actual experimental finding and ideas, I want to give a very brief overview, a reminder of the organization of the human visual system, because this is the arena where we will argue that human perception, visual perception is emerging from. And basically you can think about this system. It is displayed here in a schematic atlas on an unfolded brain. So we are unfolding, flattening the occipital and temporal hemispheres. And what you see that when you do that, the visual areas become like slabs that are arranged one above the other. This is the back of the brain. This is the front of the brain. If you recall from the first visual area, the one that receives input from the thalamus, you find neurons that are selective to low-level features, for example, lines of different orientations, Gabor patches, and so forth. As you go to more anteriorly along the areas, you get more and more abstract selectivity of the neurons until you get to this arena, which is the transition between the occipital and temporal poles, where we have neurons that are selective to entire templates of objects of different categories. Fortunately, they are clustered in categories, and in particularly, I'll use a lot the clustering of neurons that are selective to faces as an example for neuronal processing of visual images. So these areas I'll be talking a lot and showing a lot of results. These are neurons that are selective to templates of objects of different kinds, faces, places, tools, and so forth. So this is very briefly just a reminder of the human visual system. Now, when we think about what is generating the visual percept, we tend to think in a sort of hierarchical, sequential manner. This is even a textbook presentation of how the visual system works, where we have an object, let's say this frog outside in the world, it projects an optical image on the retina, and from there, through a sequence of processing stages, we end up miraculously with the image, a percept of the frog appearing somehow in our brain. I want to challenge this view and argue that it is wrong. I would like to argue that the information intrinsic to the visual system, and it is not a copy, a representation, or a processing of the optical image coming from outside. The information is available already in the high order visual areas. Now, I would like to give two sort of phenomenal or optical illusion illustration of this point before I go to the neuroscience of that. So I want to remind you of this illusory triangle, the Kanitsa triangle. And if we think about it, we can see that the visual percept of the triangle cannot be viewed as some kind of a processing or representation of the optical imaging that you are getting from the screen. Simply because physically and optically, there is no lines here. It is a flat uniform surface. So in a sense, the visual system is generating a percept that contradicts the optical image. It's not only sort of representing it, it actually goes against what is the optical image is telling the brain. You might say, okay, but this is because we remember objects that we saw before and that's how we do it. But in fact, you can actually clearly see and generate a percept of an image called impossible object, of an image of an object that simply cannot exist outside in the world. So the visual system has no problem in creating an image that is not, cannot even exist in the outside world, let alone processing or representing it in any manner. But the interesting point that I would like to discuss, and we study in our lab, is the neuronal correlates of this creative or intrinsically generated process. And I would like to discuss two experimental results from our group. One is an experiment conducted by Avital Chachami, where she studied a very fascinating phenomena. It is called the Charles Bonnet syndrome. It's a pathological phenomena happening in individuals that due to typically retinal degeneration suddenly they become blind as a result. But in subsequent to their becoming blind, they suddenly start seeing vivid visual hallucinations, despite the fact that they're completely blind and the retina is not capturing any signals. And I show you here few examples of the hallucinations in the patients that Avital studied. Some of the hallucinations are just textures, color textures, as you see here. But more interestingly, some of the hallucinations, I remind you, generated de novo, so to speak, in the absence of any retinal stimulation, actually involved formed objects like faces. There was one patient that saw a face floating in and out. These hallucinations are typically dynamic, they come and go. Others saw a face floating above a texture. So we have situation of totally blind individuals that see vivid visual hallucination. And what Avital did was to interrogate these patients very carefully about their visual hallucination. And she generated, based on this interrogation, a movie that simulated as best as possible. Of course, it's not possible to exactly simulate the hallucination, but try to simulate as best as possible the dynamics of the hallucinations. And then she ran the Bonnet patient while they were experiencing the hallucination in an fMRI scanner. And in parallel, she mapped the activity in healthy sighting individuals when they saw the simulation of the hallucination. And the fascinating discovery was the great similarity in activation during the hallucination. I remind you, in complete absence of any stimulation of the visuals of the early visual areas, and the similarity of this activation to the activation that was generated in healthy individuals that actually saw the optical images. And particularly similar was the activation, as you can see here, in high ordered visual areas. Now you could say, well, this is pathological cases. What do they represent about our regular vision? So I would like to discuss another experiment conducted by Itzik Norman and also by Simon Kuvis, in which this was done in patients with intracranial recordings of both single units and contacts from high ordered visual areas. And the experiment was very simple. It was an experiment of visual imagery. You know, we all can generate visual images in the absence of visual stimulation, optical stimulation through imagery. So the patients first saw very vivid images of faces and places during the viewing condition. And then their eyes were covered and they were closed. They didn't see anything. And they were asked to simply imagine at their own time the images they just saw a few minutes before and report into a microphone what the moment the image appears in their mind. So we have here a regular, typical sighted activation and an internally generated vivid imagery that is generated in the absence of any optical stimulation. And I want to show you just one example of a neuron, of a patient that had 16 isolated neurons recorded from in this patient. And this is the response to the presentation of one phase stimulus, Bruce Willis. So you see the activation of all these neurons, many of them, some of them did not respond to the presentation of this image. This is during viewing when you're actually stimulated by the image. Critically, when they close their eyes, have their eyes covered, and were asked to recall the different images, only when they recall Bruce Willis, you see this activation here that corresponds to the activation that happened during viewing Bruce Willis. So you see a reactivation of the same neurons during internally generated vision and externally generated vision. Importantly, along the entire set of neurons recorded in all patients, we find a significant correlation between the nature of the selectivity of the neuron during viewing and the nature of the selectivity of the neuron during a recall. And these are single neurons, but we also have similar data recording from contacts where we have groups of neurons with exactly the same paradigm. This time it was done by Itzik Norman. And here we have an interesting twist relating this recall during imagery to the hippocampus. It is known for some years now that there are very dramatic events happening in the hippocampus that are called sharp wave ripples. These are dramatic bursts of simultaneous activation of neurons. We estimate about 2 million neurons fire within 100 milliseconds in the hippocampus. So we have this very sharp burst of activity, these ripples occurring within about less than 100 milliseconds. And what Itzik discovered is the following. When he recorded from this high-ordered visual electrode the responses to the different pictures, he found that each contact responded preferentially to some set of images. For example, face selective contact will respond only to faces, and very weakly, as you can see here, responded to a set of non-preferred images. This is during viewing, as expected. I told you that these electrodes in high order areas are selective to certain object categories. Critically, when now the patients were asked to close their eyes and recall the images that they just saw, you see again the same selectivity re-emerging, but interestingly, it is centered on the burst of a ripple. It's as if the ripple plays an important role in this imagery event. Importantly for our discussion, the information, the selectivity that is supposedly driven by the eye, actually we can show is generated intrinsically, probably through some dialogue with the hippocampus. And very importantly, if you compare contacts throughout the visual hierarchy, you find that this link between visual activation from outside and intrinsically generated imagery is mainly in high order visual areas in this region of the human visual system and does not involve early visual areas. So we really see that the information for generating perceptual images is not driven by input coming from outside, but is generated intrinsically in the high order visual images. So to summarize this aspect, I would like to propose that the images that we see are not a representation of the incoming visual information. They are not representing some sophisticated version of the retina. They are based on intrinsic information that is located mainly in high-ordered visual areas. Therefore, if we search for a neuronal mechanism that underlies the formation of a conscious visual image in our mind, we cannot rely on information coming from outside from the external world impinging on the retina. We must search for this information in the intrinsic circuitry that exists in high-ordered visual cortex. So let's now explore what actually happens in high-order visual areas when we see different types of visual images. I already said that these regions are characteristics of being selective to certain object categories. And as I said, in this presentation, I will focus on phase responses. But I would like to emphasize that I would like to argue that the principles that I would like to propose are not unique to faces. I think, and we have some evidence, that they also generalize to all other visual percepts. It's simply the faces are very convenient because we find a lot of highly selective contacts that are selective to faces and not to any other object. So you hear about faces, but keep in mind that the principle that I'm trying to propose are relevant to other object categories as well. So where do we find contacts that are face selective? In other words, they are activated by face and not by other categories. As expected, you can see them here in the sort of temporal lobe, inferior temporal lobe. And on the unfolded brain, you can see them as expected in high ordered visual areas. I remind you, this is the visual hierarchy going from back to front. And as typical and found many times in fMRI, we find these contacts in high ordered visual areas in the inferior temporal lobe. Now let's look at the typical response of a contact that is phase selective recorded in this region when we analyze the response to individual phases, not to phases in general. You see this contact is indeed selective to phases and not to other categories. You see it here very beautifully. But let's look what happens to the responses to individual phases, individual images that we saw. And I want to draw your attention to two major points. First, you see that this contact responds to all phase images presented. We rarely find a phase that does not activate a phase selective contact. So their activation is very distributed, involving probably millions of neurons. Very distributed. Every phase gets some activation. However, and I think you can notice it too, the activation is not identical across all phases. Some phases in this contact, Woody Allen, produce much higher activation than, let's say, Barack Obama in this case. So we have a profile, selectivity profile, despite of the distributed representation, in which different phases activate to a different degree. You might say, okay, the distributed representation is because we are recording from contacts that have many neurons, but we find exactly the same phenomena when we are looking at the individual neurons. You see here 3 examples of individual neurons. Each line here is the response to a single phase. And you can see that there is a very different activation level to different phases, but you rarely see no activation at all to a collection of phases. So again, each neuron, each phase activates millions of neurons, but in a uniquely selective profile. We can invert the analysis and ask the following question. Before we ask what kind of activation we get for a single contact to different phases, we can ask what profile of activation we get to a single phase across all the phase selective contacts that we recorded from. And you see here an example. This is work done by Shani Grossman. And Shani conveniently arranged the activation, the electrodes such a way so the highest activation is on the left and the lowest activation is on the right. So you see, first of all, that indeed in each phase selective electrode, there is some response to this phase. However, you can also see that this phase activates in a very different manner different contacts. In other words, it has a specific pattern or a specific profile of activation associated with this phase. Importantly, very importantly, each phase, when you look at the profile of activation, these are the same electrode arranged in exactly the same manner. If you look at the pattern of activation to each individual phase, you find a remarkable thing. Each phase elicits a different profile of activation. For example, look at the profile here. I remind you, these are exactly the same electrodes, and you can see that Marilyn Monroe activated in a very different profile compared to this guy. Even more interesting, if you look at pairs of phases, you see that some pairs of phases have profiles that are very different from each other. We will call them long distance relationship between these pairs, and you'll understand in a moment why. And other pairs of phases produce very similar patterns of activation, very similar profiles. We will say that they are short distance. And you can, in fact, build a distant matrix, take all the pairs of images and ask what is the distance, how similar are the profiles of activation across this pair. Just like you do, for example, distances between towns in Israel. You can plot all the pair distances of all the towns in Israel, and you can immediately see that some pairs have very long distance, in other words, very different activation pattern, while other pairs of phases have very short distance. In other words, they activate in a very similar profile. A very convenient way to display this information is through a vector representation, a geometric vector representation. I hope most of you are familiar with it. I'll just briefly illustrate this idea of a vector representation or a neuronal space representation with this toy model that includes only 2 neurons. So instead of millions of neurons, we are talking about a representation made up of only 2 neurons. So depending of the activity that this chap is activating this neuron and this neuron, this will define a point in this two-dimensional surface where this phase lives, because it activates this neuron to that level and this neuron to that level. However, Marilyn Monroe activates this neuron very differently, so she'll be situated here depending on the activation. This is, and notice that if the activation of this chap and Marilyn Monroe are very different, then there will be a long geometric distance between them in this representation, while these two guys, because they have very similar profile, will have very short distance in this representation. This is, of course, a toy representation of only 2 neurons. In reality, as I said, each phase activates millions of neurons, so we have a space made up of millions of dimensions in which these different phases live, activation space. We will call them neuronal shape space or image space. Now, you might ask, why all this exercise? Why is it interesting, and why do I bother you with all this analysis? This, I think, will address a very profound question that might sound maybe silly to you, but once we give up on the idea that the phase that we see is driven by the outside world, we are left with the following question. We know that this, this phase is associated with a specific pattern of activation, this profile. But the question is, why this profile? What is the logic, or what is the reason that this specific profile is eliciting this phase and not another phase? Why this profile might not list an image of a frog? Why specifically this phase? What is the information? What is the rule? What are the rules that determine that this pattern will be leading to the percept of this image in our mind? And here I would like to get inspiration from the very original and deep ideas of the structuralist school, and in particularly the Ferdinand de Saussure, who is one of the most original pioneers in the field of semantics, who actually addressed exactly the same question in the field of semantics. And the question is very simple. What is the meaning? What gives a meaning to a linguistic concept? For example, tree, mountain, okay? What about the word mountain gives meaning to our concept of mountain? And what de Saussure proposed is the very deep idea that the mountain is defined not by the word mountain, but by the similarity and dissimilarity of mountain to other concepts. So mountain is very close linguistically to a hill, but very, very different for a long distance from the concept of, let's say, a valley. So by what de Saussure suggested is that the network of relationships, similarity and dissimilarity relationship between a pattern and all other patterns is what gives the meaning to this concept. I would like to immediately to borrow it directly, this concept, to our field of neuronal spaces and patterns. And I would like to propose that the reason that we see this phase as this phase is because of the similarity relationship and the similarities between this phase and all other phases. So this phase looks the way it is because it is very far away from this phase and much closer to this phase. So it's not the unique pattern of this phase that is important. What's important is the relationship, the network of relationship of this phase to all other patterns. And just to illustrate how powerful this concept is, I want to show you that this idea will mean that we can change completely the activation pattern of this neuron, either in strength or in its shape, and still retain the perceptual, the exact same perceptual experience of this phase if this idea, a structuralist idea, holds. So you see, this is the original neuronal space. We have this phase activating this neuron this level and neuron this level. But what will happen if we simply move the axis closer to this cluster of images? Notice that now this phase activates much less, much weaker activation. Nevertheless, if the idea is correct, we will keep seeing this face exactly as it is, because nothing has changed in the relationship, activation relationship of this face to all other faces. It's just that the overall activation changed. Even more drastically, if we rotate the axis, we have a completely different pattern of activation now generated by this phase. Nevertheless, the perception will remain exactly the same because the relationship to other patterns remains the same. So you see how powerful it is. It takes the edge out of the claim that what counts for perception is the selectivity of individual neurons and moves the information into relationship between patterns. Now, you might say correctly that seeing a face is very complex. Being conscious of or any image has many aspects to it. And in faces, this is particularly clear and easy to describe, because we can think about two aspects of conscious perception of a face, especially familiar face. The first aspect is simply how the face appears to us as a picture without knowing who the person is. In this kind of space that I'll call it pictorial space, these two faces will be short distance because they're very similar in shape, in features and so forth, in the hairline and so forth. And these two faces will be very, very different because they're very different in shape and so forth. So this is in the space that sort of represent or generates the pictorial picture of this face. However, knowing that this is Woody Allen, there should be another face, another space that is interested not in how this face appears, but who is the person. We'll call it identity space. And in this space, these and two faces become almost at the same point because this is the same individual. So in terms of identity, they should be very, very close, while these two faces, because they are different identities, will be at a long distance. And I would like to propose that in many cases, these different spaces live in different brain regions of the visual system and maybe higher order areas as well. So what kind of experimental support do I have to this structuralist hypothesis? You might say, okay, this is some fancy thoughts, maybe works in linguistics. Who said this works in the visual system? And indeed, this is not easy. And we are just in the beginning of trying to sort of find support for that. But I want to give you two experiments that I think go in this direction. One experiment, again, conducted by Shani Grossman, he had the following logic. We know that recently there is a huge excitement about the development of artificial neural network generated in computer that were inspired actually by the hierarchical organization of the human visual system. I hope I don't need to get into many details about what deep convolutional neural networks are. These are artificial networks made out of artificial neurons that are arranged in layers, and the layers actually are arranged hierarchically. The neurons are connected to higher levels in a feed-forward manner. In other words, there is no returning or inside reverberation. The signals flow only feed forward. And amazingly, if you train the connection, the strengths of connection by showing and correcting these connections by showing millions of labeled visual images, this artificial network somehow on its own learns to recognize images, identities of individuals, Barack Obama, Shani Grossman, any other name you want, they'll be able to recognize at a performance level that is better than the human performance. So we thought, okay, if we believe that this structuralist approach, this idea that what matters is the distance matrix between pairs of faces, of images, if it's really important, then maybe we should find it also re-emerging in the artificial network. In other words, if it's really powerful and important aspect of vision, then despite the fact that this is completely artificial, generated, in silicon and so forth with different architecture, this architecture should emerge maybe in artificial network. And importantly, we can look for this sort of neuronal space at each level of the hierarchy and find out whether it is located in specific layers of the artificial network. So how do you do it? I told you already how you generate a neuronal distance matrix. You simply show all these faces to a patient and look at the activation pattern in high order visual areas, and you measure these distances as we discussed. You can do exactly the same thing with artificial network. You can feed all the faces that the patient saw to artificial network, map out the activation of the artificial neurons at each level in the hierarchy of the network, and generate a similar matrix for each level of the hierarchy in the artificial neuronal network. And the question is very simple. Will we find similar architecture for the same faces in the artificial network that we found in the neuronal brain, in the human brain? And the answer is decisively yes. And the result is actually interesting. So what you see here is the similarity of the distance matrix in the human to the computer along the entire set of layers in the artificial network. We know that this layer at the bottom is interested in low-level features, just like the human visual system. We know that at the top, there are networks that are interested in the identity of the person. And notice that the correlation that we find between the brain and the computer are in the intermediate levels that are not strictly low level and not strictly high level. We can also explore this network, these layers, in a more detail by feeding the artificial network not only with the original images, but by manipulated images. For example, we did a low-level manipulation. For example, we removed the background from the original images, fed them into the artificial network, and asked, will that destroy the distance matrix? Is the neuronal shape space of the artificial network sensitive to this manipulation? More interestingly, Shani also did identity preserving manipulation. In other words, take for example the image of Barack Obama. It is still Barack Obama in all these images, but she rotated them into a different view. So if the layers that are correlated to the brain are identity recording layers, they should not be affected by this manipulation. However, if they're interested in the appearance of the image, then the correlation should be destroyed. And indeed, that's what she found. The low-level manipulation didn't do anything to the correlation. These high-order layers do not care about the low-level features. But the manipulations of viewpoint that kept the identity but changed the appearance destroyed the correlation between the brain and the computer. So we see here two things. First of all, what we call convergent evolution. We have this amazing coincidence that two completely different systems, the human brain and neuronal network, suddenly develop the same neuronal shape architecture. We think it is a telling sign that this is indeed a very important aspect of human visual processing. And the second one was the fact that it points to high-ordered visual areas to be the site that is interested in the appearance of images, not in the identity of the images. I can discuss later where we think the identity neuronal space lives in the brain. Another more direct prediction that should be coming trivially from what I said so far is the following. If you have two images that appear to you to be similar to each other, by the way, these are not the same person, this is Obama lookalike. If they are similar to each other, then the prediction should be that their neuronal distance should also be shorter. If, however, you have two images that are very different from each other in appearance, then the prediction is that their distances in neuronal space will be longer. In other words, that they will activate far larger, far more different profiles of activation. This can be tested experimentally, and this was done by Ido Davidescu, who simply compared perceptual estimation of distance. In other words, he asked subject to estimate how similar or different pairs of faces are. For example, these pairs of faces were estimated, as you can judge for yourself, as very close to each other in appearance, while these two faces were judged as very different. So therefore, they live in high levels on this perceptual distance scale, while this images live at very low level in this perceptual image scale. In addition, these faces were presented to the patients and their intracranial recordings, so we could get the neuronal distance. How similar is the activation pattern for these faces and these faces? And you can see in very nice correlation that these similar faces have long distances in neuronal space, and the similar faces have short distances in neuronal space. Very importantly, you might argue, this is because of low-level features and so forth. So in a very corresponding manner to what we found in the case of the artificial networks, it seems that early visual cortex corresponding to the low layers in the artificial networks do not show any relationship between the neuronal distance and the perceptual distance. So this again argues against the involvement of early visual cortex in the generation of a perceptual image. And so what I would like to propose is the pictorial space, the appearance of how we see images, lives in high order visual areas, not in low order visual areas. The person identity space, we think, lives in the medial temporal lobe and trianal cortex hippocampus. This is based not on our work, but the work of Rodrigo Quiroga and Itzhak Fried. And I'll just briefly mention the reason we suspect that the identity lives there is because they discover neurons that are completely invariant to changes in viewpoint of a person as long as the identity, in this case Jennifer Aniston, was maintained. So there are neurons that don't move in neuronal space if you change completely the appearance of an image as long as the identity is preserved. So we think, and based on this data, very elegant data, that person identity, the recognition of who you are, lives in medial temporal lobe structures. So the interim summary that I would like to suggest here is that conscious visual images acquire their appearance and identity through the relations of the neuronal activation patterns to other image-related patterns. So what really matters is not the unique pattern of activation of each image, but the network of relations by which each image is both determining and being determined by the distances to other images in the same space. So single neuron activity profile, something we were studying for years and being focused on for years, actually is not the defining aspect of perception. Of course, it is very important as a part of generation of the pattern, but what really matters is the relationship of patterns of activation, not even the multivariate pattern of activation itself. Well, I would like to propose that the appearance of an image in our mind is defined by high order category selective visual sites, while the identity, who the person is, what specific car you recognize and so forth, is likely defined in medial temporal lobe structures that show invariance to the appearance of images, but maintain sensitivity to the identity. Finally, in the last part of the talk, I would like to raise another very, very deep question that again comes from the insistence that the images that we see do not come from the outside world. They are generated intrinsically. Now, so far what I told you and what we all do as scientists is a story of what I, the scientist, found looking down on the brain of the patients or even of myself when I'm scanned in the magnet and so forth. But this is my view from outside of the neuronal processing happening inside the brain. The question that is generated, the neuronal mechanism and the aspect that is generated is in the brain, has nothing to do whether I'm looking at this pattern or not. The fact that I decided that this is a pattern has nothing to do, does not tell the brain anything, that this is indeed a pattern that should be taken as a meaningful pattern. So the deep question here is what, from the point of view of the neurons in the brain itself, what defines a bunch of neurons that are firing collectively as a meaningful pattern or assembly of activation. And if you think about it, if the hypothesis that our perception of, let's say, a face is completely determined by the relationship of patterns of activations in this neuronal space, then obviously you will not have an appearance of a face if you don't have a neuronal space or if you don't have a meaningful pattern. So if the mechanism that defines this neuronal activity, collective activity, as a meaningful pattern does not occur, does not appear, we should not be able to perceive consciously this image. In other words, what I like to say is that the mechanism that binds this neuron into an assembly of meaningful assembly is the same mechanism that moves this image, optical image, when it appears on our eyes, from the non-conscious state to the conscious state. So maybe an avenue to find out what is the mechanism that binds the neurons together should be by searching what happens in the brain when a face moves from being unconscious to being conscious. And the way I want to present it is from some results in our lab that were done over the years, first by Karlnit Green-Spector and then by Lior Fish, where we created a situation where the same visual target, this face that you see here, is sometimes seen consciously and sometimes not, although it is presented in both cases to the eyes. And the way you do it is by presenting this image in a paradigm called backward masking, presenting it very rapidly, so rapidly, and then immediately follow it with a very high contrast mask in a way that if the mask is too close to the target, it turns out there's not sufficient time for the brain to process the target, and what the patient or the subject reports is seeing only the mask. They don't see the target. However, if I move the mask a little bit and leave some room for processing for the target, then suddenly the person will say, aha, now I see the face. And by titrating very carefully this time, I can create a situation where the same optical stimulation sometimes elicits a perception of a face and sometimes only the mask. And now we can do this experiment, and we did it in the patients in intracranial recordings of face selective contact, just like the ones that you saw before. And let's ask what happens into these contacts when the person is saying, aha, I see a face, versus to the situation which is the same physically when the person says, no, no, I don't see the face. And there is a striking change in activity in this contact that you see here. This is the stimulus. You see the target face was presented very briefly to something like 30 milliseconds, and then the mask. The entire event happened for about 100 milliseconds. And look at the activity. This is the activity when the patient said, oh, sorry, I don't see the face, only the mask. And this is the activity when the patient said, ah, I see the face. Two major effects are evident. One, you see a big amplification of the signal when the patient saw the image. But even more interestingly, you see that despite the fact that the image was quenched by the mask within 100 milliseconds, the activity ignores this quenching and continues to reverberate for hundreds of milliseconds, something like 300-400 milliseconds, despite the fact that the presentation was only 30 milliseconds. This, we argue, is a signature of a very powerful process of recurrent mutual activation of the neurons representing this face. These neurons are activated by the face in this case, but you can take the face away and they will continue to activate each other in a positive feedback loop continuously until, likely due to adaptation, the activity will go down. So this is a typical signature for reverberation recurrent activity where the neurons are activating each other, eliciting the activity that we see here in the contact. And notice that this difference between the reverberation and non-reverberation is the differential aspect between seeing the face and not seeing the face. So what I would like to propose to bind these two ideas is that the mechanism that joins all the neurons that are participating in making this face appear in our mind are bound into a coherent pattern or coherent assembly, or to say it in the words of the neuronal shape space, the different dimensions of the space which were scattered, unrelated to each other before the recurrency happened, are bound together through the recurrent positive activation of the neurons. And this process of recurrency is what is generating transiently the neuronal shape space that allows this face to be expressed and the distances of this face to all other faces to be sort of measured or materialized and so forth. So the formation of the neuronal shape space through recurrency is the factor that makes this face appear in our consciousness. And you ask, is there some evidence from anatomy, from what we know about the structure of the cortex, that supports this recurrent idea binding through recurrency? And there's ample evidence. I started my career as a neuroanatomist, and looking at the cerebral cortex in anatomy, high resolution anatomical image, you are struck by the massive and dense networks of lateral connection. This is unique to the cerebral cortex that join together neurons that are situated near each other. These are excitatory lateral connections that form ample basis for recurrent activity, and the strength of connections, of course, might be the circuit that holds the information that is necessary to give us the appearance of different images in this neuronal shape space. So the aspect of this connectivity is to generate the neuronal feedback and to generate the specific patterns that are associated with the appearance of a specific pattern. So to conclude my entire talk, I would like to propose that the conscious visual percept is the outcome of intrinsic high order cortical mechanism, not a presentation of the external visual stimulation. The early visual cortex is very, very important, of course, during regular vision to help us select the proper intrinsic activation, but it is not necessary. The information is already located in full in high order visual areas. As of course we experience also during vivid dreaming, when we have vivid visual images when our eyes are completely closed. I would like to propose that the visual percept appears in consciousness when a group of neurons is bound through recurrent mutual activation, leading to the transient formation of a neuronal space in which this pattern becomes meaningful through this binding of recurrent activity. And inspired by the structuralist school, and particularly the work of de Saussure, I would like to propose that, similar to the acquisition of semantic meaning, the acquisition of appearance of a conscious image is determined not by the unique activation pattern of this image, but by the network relationship of this pattern to the activated pattern to all other patterns in the relevant space. So thank you, and I would like to acknowledge the students that participated. I mentioned them along the way: Itzik Norman, Shani Grossman, Avital Chachami, Ido Davidesco, Michal Arel is the manager of the lab, and Simon Kuvis and Ashesh Mehta and Itzhak Fried were a collaborator. Thank you, and I think we can, I'm happy to answer questions if there are any.
Rafi, I have a question.
Rafi. Maybe we can all unmute ourselves to give a big applaud for this interesting and intriguing talk. And I'm happy to suggest that everybody can ask questions. Please unmute yourself, or you can write in the chat and I will ask Rafi for you, but you can. So Uri, did you have a question?
I have a question. Basically, Rafi, basically to get this assembly, you need learning, basically. After you get learning, you get this internal representation. And basically we know for many years, when you do imagery, you go down even to V1 to activate. This is the study by Alumit, for example, that.
I know, but I disagree with this study. Anyway.
Okay, disagree, but this is what basically people show, that when you have imagery or dreams, you go down to the neuron that already saw the vivid, the real images. So basically what I'm trying to say, that once you learn something, you have your internal presentation. And when you want to actually do imagery or think about it, you activate this network that you did by learning or memory. And this is actually what you have by sustained activity. So you need time for other to go top down or anything else for this network. So don't, okay.
Great question. I completely agree with the first part. This is basically formed by learning. Furthermore, I will argue you can change a neuronal space through learning. You know, we have neat examples of, let's say, adaptation effects that are distorting the shape. For example, think about what's called the other race effect, where, you know, faces that don't look like individuals suddenly become, you can start distinguish between them. I will argue that these shapes are definitely formed by learning. Interestingly, even an artificial network that learns builds up similar spaces. So this is interesting. I completely disagree with you based on experimental findings with the notion that you need in order to see activation of early visual cortex. I disagree with the results of Alumit Shai. We have opposite results. Furthermore, there are fascinating cases. You know, we need sometimes to dig into old literature when we are asking these questions. And I was interested in hallucinations and Bonnet syndrome, and I realized that there are many reports of people developing very vivid hallucinations after massive destruction of early visual cortex. You have patients with the entire occipital lobe destroyed and yet reporting very vivid hallucination. This was repeatedly reported in the clinical literature, but somehow forgotten. In our hands, when you are trying to image, you actually suppress early visual cortex. And also I showed you results from intracranial recordings that show that we don't see any effects of vivid imagery in early visual cortex. Udi?
Can I ask a question? Can I ask a question?
Sure.
Sharon, are you taking the questions or should I?
Yeah, sure. Yeah.
In the EEG experiment, the last one where you said that they see the face or didn't see the face, how can you be sure that the recurrent activity that happens after the main peak is actually of the face and not anything to do with the mask? Because presumably they can see the mask. And if you present the mask on its own, what kind of activity do you get?
Excellent question. And I mentioned these contacts were phase selective. And in fact, one of the major findings about high order category selective areas that are very, very poorly responsive to mask, to textures in general. And this is, of course, the first thing you must have in order to study this kind of experiment. You must rule out the possibility that the mask itself is activating. And the mask itself is poorly activating these neurons. And you cannot account this reverberatory activity to the mask. Furthermore, I mentioned only one experiment because of shortage of time.
Yes.
We more recently have beautiful results from ecological vision, where we found during natural viewing that it turns out that you get exactly the same activation in these phase selective neurons regardless of time of fixation on a face of a patient. So a patient can look at the face for 100 milliseconds or 300 milliseconds, and you'll get exactly the same profile of activation regardless of the input. So again, we think this is evidence for recurrent activation that sort of does not depend anymore on the input in order to generate activation. I see Udi Zohari is asking, so let him give him. Udi, are you?
Thank you, Rafi. Yeah. I was wondering, First of all, wonderful talk. I really enjoyed it, and I'm
That's why I asked you
intrigued
to talk.
about your sort of. I'm intrigued about your sort of binary division between networks that have to do with identity and pictorial information. Just look at the picture that you have now. I can think of many, many ways in which I will sort of, the distances between the identities of people will depend on the question. For instance, if I were to decide about their age, the distances would be 1, okay? If I were to decide about their familiarity with me, the distance would be different. So I can think of, and I think there are many, many ways in which you can sort of look at the same images and sort of reconstruct the relationships between them. So do you think that for each of these ways you have a different representation? I mean, this is it's a problem, I think.
It's a beautiful question, and I completely agree with you that is something that we need to think about. The way I'm thinking about it these days is the following. There are major subdivisions. For example, recognizing Udi Zohari versus the face live in different regions. Looking at different aspects of Udi Zohari, I will argue, involves enhancement of activity of the pattern, changing the pattern of activity within a specific neuronal space, probably through attention. This needs to be studied, and it needs to be studied to see how the distant matrix that I showed you might differ if I pay attention only to your nose, let's say, only to whether you're smiling or not. By the way, there were attempts to explore representation of expressions versus appearance of faces, and they do show some segregation. So it's a beautiful open question for future study. We are just, the proposal is just the beginning, and of course there are many, many follow-ups that need to be done.
Can I ask a question?
Yeah, sure.
The question is, if you analyze the data individually for each patient, do you find the same space? And I guess you do not. How can you then find the correlations with the CNN?
Excellent question, and of course that was one of the reviewers.
Not me.
Yeah, you're a good question. Not you, but it's definitely a valid question. I of course put under the rug the fact that because these are patients and we have few electrodes, we generated the space from by assembling many electrodes from different patients, which in fact the fact that it worked, as you noted, is already telling us something interesting about maybe the canonical nature of representation. By the way, the result that I showed you about the relationship between perceptual distance and neuronal distance were also done in different sets of individuals. The perceptual distances were done in students at the Whiteman Institute. The patients were in Long Island, and yet we find this correlation. But fortunately, we had few patients where we did have a number of electrodes in each patient, so we could construct the neuronal space in individual patients, compare them to the deep neuronal networks, and the results were largely the same. We didn't find major effect. I suspect that if we could get a lot of data from individual patients, what I told you will only be intensified. In other words, the weak correlation, the noise correlation that we found between perception and neuronal activity might become much sharper if we had much more data. But of course, we have technical limitations. But still, it also tells us, probably through developmental constraints or whatever, that we all have roughly the same spaces. It's not that each one of us has totally different space than the others.
But isn't that surprising that for all of us, Marilyn Monroe basically stays in the same relationship to Barack Obama?
I remind you, I was talking about appearance, not about identity.
Okay. Yeah.
Appearance, I would argue that Barack Obama and Marilyn Monroe. Now, again, we are not talking about subtle differences. We are talking about major differences, and that's the
Cool. Yeah. Thanks.
Rafi, I also have a question about the role of early visual cortex if it is not really used to or not necessary to create percepts, then what is it good for? And in a way, do you think it mainly has to do with the fact that we're dynamic creatures and we need it for the action, vision for action, or do you think that it is related to the revival of these perceptual patterns, and then it ceases to play an important role? So yeah, that's one of my.
Well, it is very important in daily vision, because then the eyes are open and we use it. And also Uri mentioned it, and I agree, it might be very important in learning. In other words, in setting up the network that we see in higher order areas. It might also be related to Udi's question in the sense that it might be helping us to select aspects already at the early level. For example, if I'm interested in certain aspects in the visual simulator, not others, it might guide the selection. Spotlight of attention, you know, is well known there. So there are many, and of course you mentioned action, where we don't just perceive things, but we want to immediately guide movement. So all this, definitely, I'm not arguing that you don't need the early visual cortex, but not for the generation of perceptual images. That's my argument. Vivid perceptual images, not some vague images.
Mm-hmm. Thank you.
But Rafi, shouldn't you make a distinction between perception, which requires obviously early visual cortex, and the generation of the perceptive sense of imagination, visual imagery, which does not, of course, require V1. I mean, because perception is of what's out there in the world, right? I would think. That is how I see it. The fact that you can activate it, you do not need the outside world in order to generate a vivid image in your brain, does not mean that this is perception. I would not call that perception. I would call that imagery.
And what about the hallucination of the Bonnet syndrome patients, for example? What do you call? They say they see pink rabbits jumping up in their room, up and down. You know, they are so worried about it. It's a very important sociological and cultural aspect. Many of these patients do not report their hallucination because they are so vivid, they're afraid that they're going insane and starting to be psychotic. So you're talking about a person that sees things so vividly that they just look, you know, as if they are there, but cognitively, of course, he knows that this must be hallucination. So I think I will call this perception. I mean, this is semantics, you know, you can call anything as you want, but in terms of if we are interested in what generates a vivid visual image in our mind, call it perception, call it imagery, I don't care. I will argue you don't need early visual cortex for that.
Well, I would argue that you need V1 to live, okay? Because you need to understand what's out there in reality, right?
Sure. No, I wouldn't give up on my V1.
Can the syndrome exist amongst those who are blind from birth?
No. The syndrome is actually an analogous of phantom pain and tinnitus. It is a case of the quick transient denervation, where probably the hypothesis is that if you deprive the high order areas from their input, they become oversensitized, and then, for example, noise pattern might activate. We actually have data in our study with Avital that maybe noise, spontaneous fluctuations are actually driving this hallucinations. I didn't have time to get into it. But typically, if you are blind from birth, who knows what you are experiencing actually? You know, talking to blind people from birth, they sometimes say that they see. And who knows, maybe they have some kind of perceptual images. I don't know. They don't have any developmental aspect. But maybe you are born with some structures. It's difficult to get into the mind of people who are blind from birth. You know, what do they actually experience?
Micah, yeah.
Hi, yeah, thank you very much. I really enjoyed the talk. As someone who's really interested in representations, maybe that terminology kind of reconciliates the idea of pictorial, input and internally generated activity and perception. Yeah, so I think the idea of representations and what you mentioned, the experience dependency of representations, is also really important in your thinking of how the face space or an object representational space is warped by different parameters such as experience. I'm really intrigued to know whether you have any formalization of how these representations are warped within one subject as a function of experience. So what is the time scale on which, say, my representation of a previously unfamiliar face changes as a function of exposure? Do you have any evidence on a single subject level and over time how to describe that?
I have suggestion. First of all, again, it's a philosophical question, but when you talk about representation of what? What do you have in mind? Because my problem with representation is typically people say, like Udi said, representation of the external world. We see something and we have a representation, but I think I would like to argue against it. I think now, if you're talking about the representation of distances between patterns, I'm completely with you.
Yes.
Now, about the timing, there is phenomena that I agree it's completely speculative. One powerful prediction of the structuralist idea is the following. If I distort the shape space by changing something that is not the image that I'm seeing, it should change the image. Okay? So we have a beautiful example of that, although of course you might have many different interpretations of it in a phenomena called adaptation. For example, this beautiful, maybe I have it here just to illustrate. Can I still move my...
You're still controlling the screen.
Yeah, so maybe I have it. Okay, you have this phenomena of adaptation. You see here a morph version between Obama and Bush, and you can select where you see an image that is both Obama and Bush. And maybe it's not now the time to do it, but if you look for a long time into the image of Obama, remember, you're not looking at this intermediate, you're looking at Obama, you will notice that being exposed to Obama for a long time changes the face here, which is not Obama, it's some kind of a mixture, to look more like Bush. Now, of course, there are many interpretations of that, but one interpretation is that you distorted the neuronal space, so you moved the activation, the distances from here to here because of the adaptation, and because you change the space, that also changes the appearance of this image. So adaptation takes about 30 seconds or 1 minute, and you already change the neuronal space. If this is, of course, what is happening, there's a big if here, which will be beautiful to study, but again, this is future studies.
Yeah, I think this is much more going into like the short-term visual adaptation-based representational warping. And I say representations because I think that it combines both the top-down and bottom-up aspects of vision. So that's how I see what a representation is. It's determined based on the input, but also based on an expectation and whatever I'm imposing onto the visual input, I would say. And so I was more, sorry, maybe I wasn't clear in my question. I was more thinking about like how actual visual and personal semantic familiarization with an identity or an image or a person would actually change the long-term representation. So like how does this face space or the location in the space change not so flexibly as with the phenomenon of visual adaptation, but in a more long-term meaningful way? Like why is one famous face closer to another famous face, and what has to happen, and how long does that take for it to happen?
Well, in the case of famous faces, I will argue you are also moving into the identity neuronal space, and this is a different game. So first of all, by learning and becoming familiar with a face until it becomes familiar to you, famous to you, you are I'm not sure you are changing the appearance of this face, but you are definitely building up an entire space in the hippocampus and entorhinal cortex that represents who this person is, And in fact, Galit Yovel from Tel Aviv University has beautiful examples showing how differently we view faces that we are familiar with versus faces we are not familiar with in terms of their change appearance. So not in terms of how they appear to us, but how sensitive we are to their changes in appearance and so forth. This is typically learned by, again, exposure, long-term exposure. But I want to take take note about something you said, that you think that representation is both top-down and bottom-up. And what I try to argue in this presentation, that it is not bottom-up and not top-down, it is intrinsic to the visual cortex itself. So you can get a visual, the information about how an image appears is intrinsic. It's through the lateral intrinsic connections within high-ordered visual areas. It does not require top-down information. It does not require bottom-up information. It is all living in the same area where you are having the activations. And if you are interested in high order areas, they do another function. They do a function of identity. That's the way we view this kind of situation.
Thank you.
Rafi, have you considered the idea that maybe there are multiple ways to create these internal percepts? I mean, it could happen internally, as in imagination or dreaming or Bonnet syndrome, but also through bottom up through the visual system. So I'm thinking maybe it's not a critical component, but it could also lead to that. I mean, that would
absolutely, absolutely. I actually showed that you get exactly the same patterns of activation when you are viewing images optically in regular vision and when you are eliciting them intrinsically. But what I'm trying to propose is that what happens here is that the visual input is helping you select, or maybe, activate, for example, if you see something that you've never seen before, or imagine a purple elephant that you've never imagined before, maybe this language that came to your ears and got processed will be able to activate a combination of patterns that were never done before. And maybe the same thing when you see a completely new abstract structure, the visual system, the early visual cortex will sort of highlight and elicit a pattern of activity in high order visual areas. So obviously early visual cortex is active, selective, and sometimes instructing patterns to happen in high order areas. All I'm saying is it is not necessary, it is useful.
That is not necessary is makes more sense to me.
It's the way I think about it is in terms of information. I see Udi Zohari moving, I'm asking myself, where is the information that makes me see Udi Zohari? Because I could dream about him late tonight, and no eyes, no optics, nothing, and the same Udi Zohari will appear. That must mean that the information about Udi Zohari is not coming from the eye, is not some kind of sophisticated elaboration. It is living in my high order visual areas. That's where it lives.
But at least in normally functioning individuals, information is usually goal-oriented. So I'm seeing for a purpose. Yeah, vision for action in the majority of cases. So it would make sense that top-down mechanisms would be relevant and influential, no?
Absolutely, absolutely. I think we define it as a necessary thing. I'm trying to isolate the minimal information that you need in order to elicit a percept. Of course, auditory information gets integrated and affects your perception. We have all this illusion, beautiful McGurk illusion and so forth. So there's a rich interplay. But I'm trying to, as scientists, I'm trying to simplify things as much as possible.
Thank you, Rafi, very much for this wonderful talk. And let's all thank Rafi again.
Thanks for the great talk, questions.
Thank you very much for joining us.
It was a pleasure.
And I want to say that this will be available online, as Rafi agreed. And we are going to have next week Oliver Braddock and Jan Atkinson with us. Looking forward to that too. Thank you very much, everybody.
Goodbye.
Goodbye.
Bye, Rafi.
Bye.